Right, well, we are at excellent, thank you. We are at 11:04, so I think we should go ahead and get started. So we have plenty of time for today's presentation. Thanks everybody for joining, and I expect some people will continue to trickle in. This is the FNIH Active Post-Acute Sequelae of COVID-19 or PASC seminar series. This is our 9th presentation in this series, and I think we're in for a real treat as we have had for all of the 8 prior. You can see the previous presentations here. We started this back in October, and we've had a real star-studded lineup. All of these seminars are available on the website. There's a link that you can see on this slide, and we can make that available in the chat as well. These are recorded, as you heard just a moment ago, and are accessible after the fact on the website. Okay, we'll put the link up towards the end so that people can access the recordings. You can revisit these, and these seminars will also be collated and organized into a white paper trying to distill what we've learned about PASC and the science of long COVID. What do we know? What do we think we know? And what do we wish we knew or need to know to move as quickly as possible to helping people really struggling with long COVID. And so let's move on. I want to introduce in just a moment today's speaker, Dr. Jim Heath, and then we've got one other seminar planned for this series from Dr. PJ Utz. And so mark your calendar for March 9th, a week from today at 2 p.m. Eastern. And today we'll be hearing from Dr. Heath, and I'll introduce him in just a moment. To cover a few logistics before we get started, this presentation is recorded for transcription purposes, as you saw and heard. The recordings of these seminars are made available for review in the future. Everybody's muted during the presentations. For the discussion portion, we encourage you to use the Q&A and the chat features. The Q&A feature is a great way to submit your questions to make sure that, that our speaker sees them and that we can collect them and get them answered in as efficient a manner as possible. The chat is also available, and comments entered into the chat are also noted, and we can address those as well. Let's go to the next slide. I want to give a huge shout out and thanks to my co-organizer in this. So I'm sorry, I didn't introduce myself. My name's Josh Fessel. I'm the Senior Clinical Advisor in the Division of Clinical Innovation at the National Center for Advancing Translational Sciences at NIH. But the more important person as a co-organizer that you need to know about is Dr. Jay Rappaport, who's also on the line. Jay is Professor of Microbiology and Immunology and the Director and Chief Academic Officer of the Tulane National Primate Research Center. And he and I have been working closely together to put this seminar series together, and it has been a true delight. I think we've covered some really exciting ground, and we'll do so today as well. One more important piece of information to make sure that we mention out loud. During today's presentation, as is true for all the presentations in the seminar series, there will be no medical advice given. No medical advice should be inferred. This is for scientific informational purposes. And so that's just about enough for me. I want to introduce our esteemed speaker today, Dr. Jim Heath. Dr. Heath is the president and is a professor at the Institute for Systems Biology in Seattle. He also holds the position of professor of molecular and medical pharmacology at UCLA. He has directed the NCI-funded NSB Cancer Center since 2005. Dr. Heath received his PhD in 1988 from Rice University. He was the principal graduate student that was involved in the project that led to the discovery of C60 and the fullerenes. This work was recognized with the Nobel Prize in Chemistry in 1996 to 3 senior members of his group. He has had prominent positions at UC Berkeley, IBM Watson Labs, UCLA, and Caltech throughout the course of his career. He's received numerous awards and honors, has published countless publications, and has founded or co-founded a number of different companies. So Dr. Heath really has covered the bases in terms of the research landscape. He's a an investigator involved in the NIH's Recover initiative. And today he's going to be telling us about some of his very recent work on early risk factors for long COVID. So Dr. Heath, thank you so much for being with us today. And it's time for me to shut up and let you teach us some things.
Thank you, Josh. Let me share. Is that sharing correctly? Can everybody see my?
I don't know about others. I am not seeing slides yet.
All right, hold on.
I'm seeing in the chat that folks aren't seeing slides there either. You can try unsharing and resharing. We can also, perfect, yep. I was just gonna say, Trey, if you're able to share the slides, then Jim, you can control them.
Dr. Heath, yeah, I have your slides and you can actually press through them from my screen sharing if you prefer that.
I am showing Dr. Heath as frozen.
Am I still frozen? Can you see
Nope,
me?
no, I can hear you now. I can hear and see you now.
All right, I'm sorry. I've got a Zoom all day, and then when it comes to a critical point to do a presentation, it freezes up. And I'm trying to just give me a.
No talk is official until we have the tech glitch.
Okay, so am I sharing screen now?
Now I see your slides.
And then showing presentation mode or in not?
I am not seeing presentation mode.
How's that? Presentation mode, is it working?
Nope, I just see started screen sharing. Do we want to try having Trey share the slides and let you control them, Dr. Heath?
And I'll just do the next slide. I don't know what.
All right. Trey, are you able to share the slides, or does Dr. Heath need to stop sharing first?
There we go.
Dr. Heath, will you see if you press forward, if it goes to the next slide?
I can't, I don't see it going forward.
Okay. Oh, I'm sorry. Will you press the down button? No luck.
Down button. Are you sharing the screen now, or am I sharing the screen?
I'm sharing the screen, but I've assigned you slide control. So you should be able to control the slides. Yep, I didn't do that.
I can. Okay. All right. Okay.
Excellent.
Sounds good.
Excellent.
Perfect. Thank you.
Yeah, I apologize for that. It's living in the digital age. Okay. So with that having gotten past us, thank you for having me here. I appreciate it, Josh. There's many folks involved in this study. Many of you who you may recognize, a lot of immunology luminaries here, and they've been critical because this is a complicated subject that we've been trying to address. And so I'll get disclosures out of the way. And I think I don't need to describe what long COVID is, but I think it's worth noting that there's a host of other long diseases, and so the opportunity to study long COVID, I think, is relevant to things like chemo brain or post-acute Lyme, many of these chronic diseases for which vague symptoms tend to dominate, and treatments are relatively sparse. In our study, what we've tried to do is to follow patients over time, and through collecting their symptoms that they are experiencing at convalescence after recovery from acute disease, to identify, are there risk factors that anticipate long COVID, and are there immunological signatures that we can associate with those risk factors? And one way to think about this is, if these risk factors have they sort of have the flavor of being causal, although we can't prove that, but can we begin mining these risk factors for potentially therapeutic opportunities? So what are the risk factors? At what point in the disease course can we detect them? And are they independent or are they related? To answer these questions, we did a very deep phenotyping of a large patient cohort and we're looking only at 3 time points. So I'll talk about, caveats of the study, but one is that we only go out to about 2 to 3 months, which is when patients have recovered from the disease. They have developed immunity against, in terms of B cell responses against the virus, but it's not 6 months or 12 months. So there may be symptoms that emerge or symptoms that dissipate after this time point. But at this time point, we have pretty comprehensive data on these patients. And so I'm not going to go into the great detail of all of this stuff, but what I will do is, as I think comprehensive immunophenotyping can be a confusing subject. And so as we look at one aspect or another aspect of these patients, I'm going to try to use these sort of symbols so that you understand out of what type of data we're pulling our conclusions, our analysis, our insights from. Okay, and so you'll see that as we go forward. So these are the from electronic health records and symptoms. So we collected symptoms from our patients and validated them through electronic health records. We see trends that pretty much follow what has been reported in a number of epidemiological studies. We just show one epidemiological study here. Not all these studies really agree with each other in detail, but they almost all agree with each other in overall conclusions. So fatigue is very common. Cardiac issues and GI distress are a little less common, and neurological and other respiratory viral-like symptoms are somewhere in between. And when you look at data like this, we also can parse it out for patients that had respiratory support during the acute phase of disease and those that didn't. And there is a difference, but it's not a dramatic difference. In other words, as many people have reported, you can have relatively mild COVID and have long COVID symptoms. And all of the data I'll show you, all of the calculations I'll show you, we do correct for age, for sex, and for disease severity. And we do it multiple ways. I'll just show you one way that we've done it, but multiple ways. And this is all published in a Cell paper that I think actually comes out in print form tomorrow. So if we look at just the clinical labs and these symptoms that we collected from our patients, there's a couple of things that pop up. The most dominant one is diabetes and type 2 diabetes. And I think the reason why that we identify that, it's a real relationship between type 2 diabetes and long COVID, but we also have a lot of patients with diabetes. And so just statistically, we're able to resolve that as an important comorbidity for anticipating PASC. And there are other things that pop up like congestive heart failure. That's not exactly shocking to see this kind of stuff, but these are relatively few patients. And so we don't really have the bandwidth in our data to study these kind of anticipating factors in high depth. But I would say diabetes is pretty firm. We must have 40 or 50 patients that are diabetic in our cohort. Oh, I'm sorry, I'm flipping the slides in the wrong way. Okay, so one thing that we did, and really in response to things that we've been hearing from patients, is that patients will oftentimes feel they'll have long COVID symptoms that may be fatigue, they may be memory problems, what have you, and they were having a hard time getting attention from their physicians. And so we tried, one of the first things we tried to do was, can we find biomarkers that associate with particular adverse sequelae that patients are experiencing that would help define the disease a little bit? And I think the concept is, if you can define the disease, you can begin to treat it. And so we mined the plasma for protein or metabolic markers that we could associate with specific PASC. And these measurements in the plasma now are carried out at the same time as the symptoms are being reported at this convalescent time point, 2-3 months out. And in fact, there's not a lot there, but you can, in fact, find things that are pretty statistically significant, just not a lot of things, considering the depth of our database. So for neurological PASC, we found evidence of an altered circadian rhythm, and that was, there's a total of 4 proteins involved in this particular circadian rhythm cycle. 2 of those proteins we were able to measure in our like 500 and some proteins that we measured, and they both sort of reinforced each other in saying that the circadian rhythm is disrupted. It's actually very significant. And I would say that's really the only proteomic or metabolic signature we found for a neurological PASC, but it was pretty significant. For respiratory viral PASC, which is, we're looking at, in both neurological PASC, we're looking at a collection of symptoms, and for respiratory viral similarly, we found, and this has now been reported, I think, elsewhere in multiple places, we found repressed cortisol and cortisone levels. Now this is, known in the medical literature as Addisonian crisis or acute adrenal failure or things like this. Is typically treatable through cortisol replacement therapy. So that's certainly something that I would suggest could be explored. But the symptoms are in many ways very similar to what one sees in some of these long COVID patients. Now, a caveat with seeing low cortisol or low cortisone is that patients that are on steroids are often experiencing also low cortisol or low cortisone, and it's simply because the steroids and the cortisol molecular structure are very similar, and they can compete with each other in the sort of regulatory cycle. And we, in fact, at acute disease or early in disease course, so this is at diagnosis, T1, T2 is at acute disease, T3 is at convalescence. Early in the disease course, low cortisol does correlate with steroid treatments, but not at all late in this stage. And so this seems to be an independent of steroid treatments. Okay. So earlier, I think about, middle of summer of last year, there was a report that patients that were antibody positive for EBV, meaning at some point in their life they had EBV and developed antibodies against it, were more prone to having long COVID symptoms. And so we looked for direct evidence of this. And we looked for direct evidence not just for EBV, but also for cytomegalovirus, because these are the 2 most common latent viruses in our populations. And the difference in looking for direct genomic signatures of these viruses as opposed to antibodies is that those genomic signatures are going to be very transient in the blood. And so it's harder to catch, but it can help you identify if you had reactivation of a virus, when that reactivation happened. And so when we analyzed for these 2 viruses, as well as for SARS-CoV-2 levels, RNA levels in the blood, what we found is that you see quite a bit of it, maybe 20% or 25% of the patients at this initial diagnosis point. It's reduced by 3 to 4-fold just 1 week later, and barely detectable at convalescence. And in fact, if you look at the SARS-CoV-2 RNA-emia, we find that associated with patient mortality, which was also published last year in this paper here. We do not find any evidence for CMV reactivation, but for EBV we do. And then the SARS-CoV-2 RNA-emia seems to be important as well. And so then when we take those viral signatures that we see in the blood and we associate them with symptoms that patients are developing later at convalescence, we find that by and large, EBV viremia is associated with neurological-like symptoms, a little bit with fatigue, but mostly these things are coming up in neurological. SARS-CoV-2 RNA-emia, similarly, but I would say this is really as much of a signature of disease severity as anything else. This does not associate with disease severity. But even when you correct the SARS-CoV-2 RNA-emia for disease severity, you still see these associations pop up, and that's what I'm presenting here. And so this says that having these two viruses detectable in the blood at diagnosis of COVID-19 anticipate PASC. If we looked at nasal swabs, then at the acute stage, it can be a PASC factor for taste and smell loss only. But it really is not as major of a factor for post-acute sequelae as is the viral load in the blood. Okay, so autoantibodies. This is something that clearly has been studied extensively, especially in association with long COVID, I mean with acute COVID. And so it seems reasonable that they would play a role in long COVID, and in fact they do, as I'll tell you. And so we know that the presence of certain type 1 interferon, for example, can have really impact a patient's disease journey through the acute stage of disease. But also, so this is certainly something that we try to capture is anti-interferon alpha autoantibodies. But in our Cell paper we published a year ago or so, we found this class of cells called atypical memory B cells, which are kind of a hot literature topic right now. There's a number of studies that associate them with different disease conditions. One association is with lupus, systemic lupus erythematosus, and that's prompted us to look for these particular lupus-associated autoantibodies as well. So we did this autoantibody panel, and here's what we found. And there's actually a pretty deep set of findings, and I'll just be able to give you the highlights. First, we found that I'm plotting here for 2 independent cohorts: the cohort we got from Swedish and the cohort from Helen Chu's program at UW. If you have, these are SARS-CoV-2 protective antibodies. So if you have one protective antibody, you have the other ones. That's not too surprising. Here's the autoantibodies down here. And also you see that there are weaker correlations, but still, if you have autoantibodies of one flavor, you're likely to have autoantibodies of another, increased likelihood. What's surprising is that at least for the IgGs, for the mature SARS-CoV-2 protective antibodies, in both of these cohorts, the more autoantibodies you have, the fewer of these antibodies, the protective antibodies you have. And so this suggests that perhaps people with elevated autoantibodies are more susceptible to having things like breakthrough infections. We looked for how these autoantibodies might associate with different PASC, and in fact, they do. Largely sputum, and these some of them are pretty significant, but there's a group of these here that seems to have a trend: inability to exercise, some gastrointestinal trends. But the result of this is that, in fact, autoantibodies at COVID-19 diagnosis are in fact PASC factors. And finally, looking at that lupus connection a little more, but also asking the question of, are these autoantibodies developing because patients had COVID-19, or did they pre-exist? And what we find is that patients that have autoantibodies at convalescence, by and large, have autoantibodies at diagnosis. That's what this data here shows. And at diagnosis, those autoantibodies are mature. They're IgGs. There's no IgMs. And so this says that these autoantibodies are present prior to disease. They don't really associate with disease severity here, is what this is saying. But they do definitely associate with the numbers of these atypical memory B cells. And so that motivation we had for this connection of looking for systemic lupus erythematosus-like signatures in the immunology of these patients seems to have been well-founded. And so I think there's a number of things that are relevant to point out here. Number one, if you have autoantibodies tested by someone like LabCorp, they're going to tell you if you have autoantibodies at a clinical level, which is going to be actually quite high, like 4 standard deviations above baseline. We're looking at patients that are 2 standard deviations or more above baseline. And so many of the patients that we see autoantibodies present in, they're at subclinical levels and they wouldn't be detected by a standard LabCorp test. And so I think that's important to think about. Second is that the autoantibodies tend to be functional. And so if we look at patients that really had clinical levels of autoantibodies, meaning in the top 0.001% or something like that, or 4 standard deviations above baseline, the impact on post-acute sequelae is advanced, okay? So autoantibodies definitely have a role. The more you have, the more role they play. And I think that suggests an interesting avenue for thinking about therapy. So one of the biggest data sets we collected from these patients was single cell analytics. And we did all kinds of things at the single cell level. And I'm only going to give you some highlights. But at the base of these highlights, we have T cell receptors and B cell receptors. We have surface proteins. We have single cell RNA-seq. We have polyfunctionality, etc. And all these data sort of reinforce the comments I'm going to make. So what we used, I'll just talk about T cells here. And so what we used T cells for is basically barcodes, because you can use a T cell receptor as a way to barcode how T cells are evolving over the course of a disease and recovery. You identify certain T cell receptors at initial diagnosis, and you associate those with their T cell phenotype. You see how that evolves at the stage of acute disease and how that evolves at the state of recovery. And what one normally expects to see is that you're going to have very few a large number of T cells, but lots and lots of T cell clonotypes that are really going to form a naive pool. And as infection ramps up, those are going to become largely effectors, and they should then sort of contract over time into a memory phenotype pool. And so we use these TCRs as barcodes to follow that. And in fact, what we find is that by and large, that is what happens, but it isn't what happens in all patients. And so to get a little more clarity on this, we also worked with adaptive biotech and with just public databases to say, well, can we not just say what the T cells are by barcoding their T cell, using their T cell receptors as barcode, but can we identify T cells that are specific to SARS-CoV-2 or to other viruses? And so here I'm plotting, we found like 150,000 T cell clonotypes that were in our patient population that we could assign to some antigen associated with SARS-CoV-2. And so I plotted these and projecting them onto this UMAP of CD8 single cell RNA-seq data. And then I've taken literature data of CMV. Remember, CMV is not reactivated, but we do see CMV specific proteins. I've taken literature TCRs from CMV and also plotted them over time. Okay, so if we look at this UMAP here, on the right, you've got the effector T cell populations. On the far left, you've got basically you go naive in this direction, and then stuff in between. And then this little purple group up here is the proliferating T cells. And so you see these purple T cells that are SARS-CoV-2 specific are proliferating during the course of initial and diagnosis and acute disease, but are pretty much gone at the time of convalescence. And then you see these effector populations. They may peak up a little bit, hard to tell, but they in most patients, and I will say this is statistically significant, they go up and they go back down. And then you'll see, like in the CMV, you see a similar behavior. This is a little clearer because we got more T cell clonotypes assigned, that you've got elevated levels of these T cells that diminish at recovery. But in some patients, you actually see these effector cells populations increase at recovery. And here's, in fact, and those are patients that have gastrointestinal PASC. So in other words, if you look in GI PASC early on, you don't actually see much of these, and this is now just CMV, but I could show the same thing for SARS-CoV-2. You don't see a lot of SARS-CoV-2 or CMV specific T cells, but they increase. And so as you go through from baseline through acute disease out to convalescence, these guys increase in population, suggesting that gastrointestinal PASC has a different kinetic pathway than do the other post-acute sequelae. I hope that makes sense. It's kind of a complicated argument, but it also is saying that because you got CMV and SARS-CoV-2 specific T cells here, this is probably just broad inflammation. It's probably not viral specific, and what's called bystander activation of T cells that are maybe not directly engaged with antigen signatures. Okay, one other thing that we pulled out of our single cell data that we integrated with the symptoms to provide a UMAP representation like this is that patients by and large clustered into having an immune system that you could characterize as naive-like, an immune system that you could characterize as Th1-like or Th2-like, in the sort of classical immuno pictures, with some patients that sit in between in what we call this intermediate group. And we also identified in these patients, and I think this is sort of consistent with, I don't know if I have this picture here. I don't. We identified in these patients that you could pretty much anticipate at diagnosis where a patient's immune system would end up at convalescence, whether it be Th1, Th2, or naive-like. By and large, patients with long COVID symptoms sat up here. Patients with severe disease sit over here. And if you look at, for example, autoantibody levels, you see there's very few autoantibodies down here in these sort of naive populations. They're really centered up here. If you look at patients that have viral signatures in them, they're centered mostly in this sort of intermediate regime here. And so we don't really know how to interpret this yet, but I think what it more or less is telling us is that the traditional pathways that people take through disease and to recovery they're not quite as diverse as the numbers of patients you have, but there is a fair amount of diversity to them. And it does explain, I think we all know PASC is going to be heterogeneous, but it explains some of that heterogeneity is just the nature of the fundamental characteristic of the patient's immune response. Okay, so finally, a calculation we did is to try to understand how are these PASC factors related to each other and how do those relationships evolve over time? And the reason why we asked this question is because if these PASC factors look like they're highly related to each other, maybe there's a limited number of treatments that one would think about for these patients. But if they look like they're completely independent, then there's going to be a large number of treatments. And so we utilized our multi-omic data set to establish just how related, and we did this for all the PASC factors. I'm just showing a few of them here, but for all the PASC factors, to look at these relationships. And the answer is really, really interesting. So here's the answer, and let me walk you through what you're looking at here. So here's the things that we've identified as PASC factors: diabetes, EBV viremia, SARS-CoV-2 RNAemia, these various autoantibodies. At diagnosis, there are a number of relationships, especially between the autoantibodies, but there's some down here between, say, viremia and type 2 diabetes as well, that say that these PASC factors have some interrelationships. Now you look at these PASC factors, but now you look at them a week later, and they're less related. And by the time the patients are recovered, there's basically no relationships whatsoever that you can resolve. And I think what that's saying is that you have a number, all these patients are starting off with an infection and with PASC factors that can basically control the pathway that that patient takes through that infection. But because everybody starts off at slightly different places, different ages, different sex, different comorbidities, those pathways ultimately diverge exponentially over time. And so this is probably how one wants to think about developing therapies, but this is what one sees when you look only late in the game. You see that basically everything is diverse, everything is heterogeneous. It's almost impossible to get your head around treating. But early on, you see there's actually a lot of relationships between these. And I think that that makes it something a little more manageable in terms of how you get your mind around helping these patients. Okay, so the importance of these PASC factors. So we find that EBV, viremia, SARS-CoV-2 RNAemia, and type 2 diabetes each explain about 30% of the PASC that we see in these patients. And then autoantibodies explain about 2/3 of the PASC. And because some patients had multiple PASC factors, this adds up to more than 100%. But the autoantibodies do not to be the most important thing here, at least in our patient population. And here's just a few statements about the fact that high autoantibodies or clinical autoantibodies and subclinical autoantibodies are not the same thing, but they both seem to matter for long COVID. This relationship between long COVID and systemic lupus erythematosus seems to have some grounding to it as well. And so that seems like an avenue that might be productive in terms of thinking about treatments here that have been effective that might be worth exploring for long COVID. And so the limitations of our study, we only followed patients out 2 to 3 months post-diagnosis. As Josh mentioned, we're involved in the National Recover. I'm directing the Pacific Northwest Consortium of that national program. And so us and the other dozen or so sites that are involved in this around the country are certainly going to have the patients to resolve things that we can't resolve in this study, for sure. We were limited to around 300 COVID-19 patients, and that means that, we're really not going to have a lot to say about cardiovascular PASC or about hair loss or some of the more, some of the rare or even semi-rare symptoms. And so I just can't answer those questions, but and it's like the reason why we resolve type 2 diabetes. We just have a lot of patients. And so we can see there's a common comorbidity. Other comorbidities are surely important, but we don't have the statistics to tell them, except for maybe these heart conditions I referred to earlier. We only looked at a handful of autoantibodies. So how would you design a national study of autoantibodies? I don't know. We're now doing the whole human proteome with our patient cohort of autoantibodies with Mike Snyder at Stanford. But I think that there are other studies that are probably doing similar, and they're going to we'll all meet together and figure out what would be the best palette of autoantibodies to look at. For sure, PASC is going to change as the genetic nature of the virus changes. This study was not able to capture patients from the Omicron variant, so I can't say much about that. Can't say anything about that. And we had very few patients in the Delta variant. But this is something that is going to be important to resolve. And just a plug for the national study, because that's going to have the statistical oomph to answer some of these questions that aren't resolved in this study. And then with that, I will. I'm looking for talented postdocs, so contact me if you're a talented postdoc. And I want to make sure, I think the funders, we had funding from NIAID, of course. We also had funding from BARDA, which, and all of this stuff was important in helping us, do the study at the depth that we did, and et cetera. So thank you so much. I'm happy to answer your questions.
Absolutely fantastic. Thank you so much, Dr. Heath. Lots of, I was writing down questions the whole time, but I will definitely give others a chance to hop in. I just want to say that Dr. Manetsky from FNIH has been able to join us. I want to give a shout out to Joe as well. The seminar series wouldn't happen without his work, along with Trey and the others to organize this. And so thank you to all of them again. Jay, I see that you've come off mute, and so I'm betting you have questions that are maybe along the same lines as mine, certainly more insightful. So I want to give you the floor here.
Yeah, thank you very much. And that was, Josh, and that was a great talk, and really some intriguing information. I was very interested, since you have quite a few diabetics in the PASC cohort, is there any relationship between the severity of PASC or the type of PASC with glucose control?
We didn't resolve that. We did try to look and see if there was. I think we looked for this in the Cell paper we published a year ago on basically acute COVID, and there may be an effect there, but it wasn't a strong effect. And I was kind of surprised by that, but a lot of these patients during the course of their treatment, and some, I think probably half of our patients were enrolled early in the pandemic, and the amount of drugs that were being tried at that time and the different types of drugs and treatments may have had a confounding effect. And so it could be that there was an effect there, but we just can't quite resolve it. But it wasn't. Diabetes came out very clearly as an anticipator of long COVID, but we couldn't really resolve how it affected the journey through the disease.
Yeah, any let's say predictive value of, let's say, A1 hemoglobin A1C at the start point?
Oh, that's it. I can't tell you that, but I could get back to you with an answer. We certainly could do that calculation.
Okay, that could be interesting. That'd be a predisposing factor. Thank you.
We have found, in terms of, there's some pretty novel immune cell phenotypes that pop up, and we were able to validate those immune cell phenotypes through basically single cell glycolysis measurements, because you can make predictions, and that's how you validate. And we published in that in Nature Biotech somewhat 6 months ago or something like that. And so we do find that glycolysis does actually play a role in some of the acute cell immune phenotypes that pop up. How that correlates with diabetes or not, we may not have the statistical power to resolve, but one would expect there would be an association.
Very good. While we're waiting for people to pop in their questions, I was fascinated by the role that time and timing seems to play in all this. And it came up for me in a couple of ways. And I have a couple of questions around that. The one that I'll ask first is having timing in there as a variable. It really struck me when you were talking about the relationships between the predictive factors and how those relationships seem to diverge over time. And I was reminded of the phenomenon that's seen in terms of opportunities for intervention. I was reminded of critical illness associated brain dysfunction, or ICU delirium, where what we've been able to tease out from the clinical studies is that things that happen in the intensive care unit and things that you do to try to help the critically ill person in about a 14-day window when they're in the ICU can have measurable impact on the development of acute delirium and measurable impact on cognitive function up to a year or more later. And so I wonder, do you think then that what your data may be hinting at is that that there may be a similar kind of thing happening in the continuum of acute COVID and PASC, where an intervention made early has the opportunity to really kind of bend the curve, as we say later on? And if you think that's right, like how do we get our brain around that, and what do we do with that information, do you think?
I think that is absolutely a message. So, I mean, one of the easiest ones, and I don't think anyone disagrees with this, is antivirals really early in the disease course are going to make a difference, because the two PASC factors of EBV and of course those are RNA and DNA viruses, so probably not the same antivirals. The autoantibodies also pre-exist, and they're functional, and we know from the studies that came out of Yale and Rockefeller and places, and we've been involved at least tangentially in some of those studies, that those strongly affect the pathway to acute disease, and you can know those ahead of time. So my guess is that offers an opportunity for a course correction as well. And before this pandemic, we were going to do the study of post-acute Lyme and on Nantucket. I would completely redesign that study based on the stuff we've learned here, because I do think that it's possible to intervene by acting early in the course and of disease progression. There's some things like I'm not sure what one does about diabetes or other the comorbidities that are going to pop up, but we know that all of these, like, as I say, they impact your course through the disease, and that seems like something that should be correctable. And there's not that many different courses through the disease. That was what that endotyping, those 4 endotypes that we found, basically said. And so it's not as hopelessly complex as one might think by just looking at all the symptoms people are reporting at recovery. I think it's not simple, but it's simpler than this worst case scenario that is suggested by symptom analysis.
I think a reason to hope is more than welcome at any stage of thinking and discussion about the pandemic. So thank you for that. People, I think, are still considering their questions. So I will stay with the theme of timing, if I might. And I was really intrigued by your findings regarding circadian regulatory proteins. And I wondered if there was any hint of a signal for a clinical echo of that, or if there was an opportunity to do something like, a Munich chronotype questionnaire to try to find some at least correlative data for what's happening in the clinical realm of potentially understanding circadian disruption in light of what you found with the proteome.
Well, there is a lot of, in the RECOVER study, sleep issues are pushed certainly a lot more in depth than we did. Basically, we didn't even ask questions about difficulty sleeping. Patients just offer that stuff when you're asking them other questions. And so we recorded it. But I think the National Recovery Study will resolve circadian disruption a little better. And I think it would be knowing that this disruption of this little protein network, which is really clear. It's when I did this analysis myself, it's the only thing that pops up, and it pops up pretty significantly. I think that tracking that down, it gives you a starting point for trying to track down a mechanistic picture of how that's happening. Which I can't give you insight into that now, but I can tell you how we would start trying to pull that out.
Jim, have you seen any perturbation of, we'll follow up with Josh's question on the indole pathway, melatonin, serotonin, any of that, methoxyindole pathway?
We must. It's a good question, and I would say that we certainly see that disruption at the stage of acute disease. I don't know what I can tell you about what we see at the stage of recovery. But at acute disease, we measure, I don't know, like 1,100 metabolites in these patients. 800 of them are whacked out at acute disease, and there's a massive loss of lipids. There's a lot of catabolism going on. It's a major disruption, and then they're all sort of recovering but they're so whacked out at convalescence. So it's really difficult to. And even in these time views to resolve what is, I guess, spectator disrupted or what is actually, really engaged and making the patients feel bad. I think that's where 6-month studies and longer are going to be able to shed some more light.
Outstanding. Well, we are very close to the top of the hour, and so. First, I want to say thanks again so much, Dr. Heath. And Joe, we usually start with you, so I will end with you and give you the last word if there's anything you want to add to this.
Thanks, Josh and Jay, for putting this together, and Dr. Heath for your presentation, which, as any good science does, asks more questions than it answered. But just terrific. So, but I want to thank you from active and what we've been trying to do. This is absolutely beautiful. So thank you. I thank you all for joining us.
Really appreciate everyone's attendance. A huge thanks to Trey, Larissa for making all this happen. And again, thank you, Dr. Heath. And we will stand adjourned.
Thanks, everybody.